Good morning. Good morning, everybody. How are you all? Welcome to the joint webinar by Biotecnika and Rasayanika. And today we are going to be joined by Promega scientists and experts from Promega India Pvt Limited. Please confirm in chat if you can hear me. How are you all? All good? Right? Fantastic. I can see a lot of responses coming in chat. So before we get started with this webinar, we have a quick introduction for you all. Which part of the world you are joining us? Can you tell us quickly in chat? Which part of the world you are joining us from? Okay, just let us know from which part of the country or world you're joining from. Great, fantastic. So I can see a lot of people joining in from all across the globe, and this is what motivates us. This is what exactly motivates us. Fantastic. Wonderful. Now, this is the greatest motivation one can get. So many people, Biotecnika and Rasayanika is able to reach out to so many of you, right? Wonderful. Shillong, Bangalore, Karnataka. Okay. Wonderful. So today's webinar agenda, as you all know, battle against coronavirus, conducted jointly by Promega India, hosted by Biotecnika and Rasayanika. And today, as I come to you live from Bangalore, the capital city of India, we all on this wonderful Wednesday morning of August 2020 know what the world is going through. India has crossed 19 lakh COVID-19 cases, with almost 50,000 on an average cases falling prey to this virus almost every day, right? Yes, I am great. Thank you. So in fact, this has never happened in the history of mankind, right? Now you see 6 billion people are looking all in the same direction, right? All of them are looking in the same direction, thinking in the same direction, thinking the same thing day in, day out. But then, why are we not getting a vaccine? Why are we not getting a vaccine for this virus, which has wreaked havoc on the world? Entire world is suffering. So we reached out to our experts at Promega India, right? Promega is a world-class organization, and Promega India, which is a subsidiary of Promega Corporation USA. And we asked them, how is this battle going on, and how is it at the forefront, and how well prepared are we to take on this virus? So Dr. Rajneesh Bharti and his team. So Dr. Rajneesh Bharti is the general manager of Promega India. He readily agreed to, meet you guys and conduct a webinar where Promega can educate all of us on how this battle is going on, how their products and scientists are helping the research scientists and medical fraternity. Okay, so today we have 3 experts from Promega, and they're going to help you understand this battle from the forefront. Now, before I actually pass on the controls to them, I have a couple of things to share with you all. Now, as I always have insisted, please share this webinar link with your friends and family members on social media, WhatsApp, Instagram, Facebook, wherever possible. Let's bring in more researchers so that we all can join hands in this crusade to neutralize this virus. Can you guys do that for me? Can you please share this? Yes? Wonderful. So another announcement is, if you attend this webinar till the end, we'll have a Q&A session, and we will also be giving you participation certificate from Biotecnika and Promega India jointly. All right? So promise me you are going to be there till the end. Yes? Type in the comment box, till the end. Yes. All right, great. So now is the time to pass on the controls to our experts at Promega India. Thank you so much, guys. I'll see you at the end with the Q&A session. Bye-bye.
Twenty-four by 7 to support the community by producing. Welcome, everyone. In today's Promega India's webinar, the topic for today is coronavirus detection and assay development. And the agenda would be the overview of Promega and effort towards COVID-19, overview of COVID-19, and then COVID-19 testing strategies, what Promega is doing, and the new opportunities for different research. Promega Corporation was founded in 1978. The headquarters is based in Madison, Wisconsin, USA. The numbers which I'm going to share is 2-year old, but in calendar year 2017, the revenue over was 400 million US dollars. We are about 1,500 plus employees and operating from 16 countries. There are about 4,000 products for life sciences research and applied sciences research, and we distribute around more than 100 countries through channel partners. So in COVID-19 update is that Promega is working 24 by 7 to support the community by producing some of the kits which are being used in COVID-19 testing. So the current Promega components, which we are approximately about 29 COVID-19 kits around the world. Promega provides sample preparation tools to approximately 500 clinical labs worldwide. The very interesting aspect of our current situation is that one of the kit has been approved by CDC for the emergency use for COVID-19 testing. And in India also, RNA extraction kit, which has been approved by ICMR. So we are always there to support the community. And through this kit, we are supporting most of the diagnostic lab in India where they are testing the COVID-19 presence. Thank you.
Hello everyone, my name is Nitin Kapoor, and in today's talk I'm going to take you all through the different methodologies for the detection of the coronavirus. In this pandemic time, the researchers and the scientists around the world are working to develop the investigational antiviral compound to treat the coronavirus infections. There are various technologies that has been developed so far for the detection of the coronavirus. So before we discuss the different methodologies that has been developed for the detection and for the testing of the coronavirus, let me take you all through the structures of the coronavirus. So in this coronavirus, if you see here in the structure, so it comprises of RNA as a genetic material. So the virus has single-stranded RNA as a genetic material, and its genome is very, very small. It comprises of 15 coding genes. Another component of the virus is protein. It has HE protein, envelope protein, N protein, and spike protein. Spike protein is also called as S protein that mediates the attachment of virus to the host cell surface receptor. And if we again look into the detailed version of the S protein, it has two components, S1 and the S2 subunits. So the S1 subunits binds to the host cells, and S2 is needed for the fusion with the host membrane. So here in this slide, we will see how this virus basically attacks to the body and infect the human cell. So the S1 subunits of this virus basically mediates the binding of the coronavirus to the human cells through the ACE2 receptors. And once virus inject the RNA, the cell got infected, and afterwards it start replicating and start producing more and more copies. So now again the questions come up: how to detect a person who has infected with the coronavirus. So the Promega have different products and technologies available right now, and we have consolidated all the information in this web page for various technologies, products, which is available with us for the coronavirus detections. The most prominent and the widely used technology which is used for the detection of the coronavirus is the RT-PCR based technologies. And as per the ICMR guidelines, this is also one of the most recommended technologies, and this is also the gold standard technologies in the market for the testing of the coronavirus. And as per the ICMR guidelines, there are three steps. The one is the sample collection, which is basically the collection of the nasals or the buccal, the nasal or the throat swabs. And the another step is the RNA extractions. And the step three is the RT-PCR. Where does the Promega's products fall into in this testing part? So we are pretty much compliance as per the ICMR guidelines, and a lot of our products are already approved by ICMR for testing purpose. So we basically offer the product in step 2 and step 3. In step 2, we offer the RNA extraction kits that can be used manually, and we also offer the automated platform that basically helps scientists to save the time. And then we also offer the products for the amplification, which is basically the GoTech qPCR products. Now, in a major step to counter the coronavirus crisis, the Promega India is supporting the government agencies through the automated instruments. The Maxwell RSC instrument is the compact automated RNA extraction instrument that can process up to the 48 sample in less than 30 minutes. So now we can believe in a typical 8-hour shift, up to 400 samples can be, processed by using this technology. Now, another methodologies which is available in the market is the serological test, which is available in the market for the coronavirus detections. This technology is also famous in the name of the antibody-based detections. So basically, there are two formats for this these technologies. So one is the rapid test, another one is the ELISA test. I'm sure many of you might be familiar with these terminologies. So rapid is just a very quick, fast test. It can be done as a point of care, or it can also be done in the lab. And ELISAs is one of the traditional and conventional method that can be performed in the lab. Now, the Promega also have developed one of the concepts similar to the ELISA's technology. So we are in process of launching the Promega's Lumit DX SARS-CoV-2 immunoassay kit very soon. And this technology is based on the bioluminescence-based technology. And since this is based on the bioluminescence-based technology, this is very fast technologies, and the results can be seen in very less period of time, and it is a very sensitive test. Now, since there are different approaches are available in the market. So which test to be used when? So in this graph, basically you can see here. If so, for example, now assume a scenario, if somebody have got infected with the coronavirus, now it will take around 9 to 10 days to develop an antibodies. And if the person has just infected with the coronavirus, and if we do the antibody test, the test will come negative. So now in that scenario, when to use the RT-PCR test and when to use the immunoassay test. So the government authorities and ICMR have already set couple of guidelines. So I will not go beyond that, but just to share couple of the recommendation where these tests has been used. So definitely the RT-PCR is the gold standard test that is being mostly used in the market. The immunoassay based test is used basically to check the surveillance or the epidemiology of this virus at certain places, or it can also be used for the infected or the confirmed patients to check the immunogenicity of the or how they are going to recover with the infections. Or there are certain scenarios this test is also used for the developing the certain kind of the vaccine development program, or certain the drug development program also. This test is being used. So with that, I would like to hand it over to my colleague, Dr. Manas Ranjan Sahu, who will be going to cover more depth and details about the coronavirus vaccines and therapeutic development area. Thank you.
Thanks, Nitin. Hello and welcome everyone. In this part of the presentation, we are going to talk about the products and the innovations from Promega, which has immensely contributed to basic and applied science research. Mostly, we are going to talk about those ones which are used in viral research and drug discovery. Let me first help you with the life cycle of the coronavirus so that you relate to the uses of that product in the future slide. Spike protein of coronavirus and the host cell receptors are engaged at the first place that brings about the binding of this virus to the host cell. Viral entry takes place either via the membrane fusion or through endocytosis. Once the viral genome is into the host cell cytoplasm, the cellular machinery then takes care of it to produce a lot of viral proteins. And these polyproteins are then used to make copies of RNA virus, which are used to translate or make a lot of structural and functional proteins that are used by the virus. Later, these structural proteins, along with the viral genome, are again encapsulated or packaged to make fresh viral particles, which upon the maturation are released and are now and these are now capable of doing fresh infection. So if we look at the life cycle, then it gives us a lot of hint that there are at least 5 or 6 checkpoints where actually we can focus and develop therapeutics for. Let's take a look at it. So we can design therapeutics to stop viral entry or the translation of viral machinery. Also, we can have therapeutics to stop viral replication, where you can see a lot of drugs have come up. No doubt, they are broad spectrum viral therapeutics, but yes, they need to be checked how efficient they are. Besides that, we can also check the translation of viral structural proteins. We can check viral assembly or the release. So any of this part can be targeted for designing therapeutics. So when the host cell is under viral infection, the host cell responds to it through a number of changes that can be metabolic, maybe cell health, or in relation to immune response. Promega has a lot of products, or I would say a different technology and different product line tools and techniques that can be used to answer these questions, whether it is in relation to drug discovery or in relation to the host response in relation to the viral infection. But most of these products are in the line of luminescence readout. So let me help you out with the luciferase chemistry that Promega deals with, because this is going to be the backbone of what products that we are going to discuss about. Promega deals with mostly three main luciferases. They are Firefly, Renilla, and Nanolux. Nanolux is a new to the portfolio of Promega, which is the smallest and the brightest. Besides, Nanolux can also be split into multiple subunits. They are named as large bit, high bit, and small bit. And these subunits, they have different affinity to each other. For example, the large bit and the high bit, they have a very high affinity to each other and can be used for spontaneous complementation studies. This makes high bit an important genetic reporter, and can be also, this combination of high bit and large bit is being used in many detection, in many luminescence platform, particularly for the detection part. But the large bit and small bit, they have the least affinity to each other. Thus, it can be used for facilitated complementation studies. And this feature of Nanolux has been used by Promega to develop tools to study protein-protein interaction. Well, most of the techniques that we have today with Promega, the new and the upcoming ones, are based on Nanolux, and they have been used for design new type of cell-based assays. They are designed to study protein-protein interaction. High bit, as we talked about, can be used for protein tagging system, and we are also going to talk about a new assay known as the Lumit immunoassay, and we are going to talk about how it has additional advantages compared to that of ELISA that is existingly available in field. So to start with, I'm going to give a brief about what Nanobit is about. It is basically a platform which uses two subunits of Nanolux, namely large bit and small bit, where independently each subunit is tagged with protein A and protein B. When protein A and B interact, either at the physiological level or under treatment or under any stimulus or circumstances, then structural complementation of large bit and small bit takes place, and hence there is a generation of luminescence upon substrate addition. The picture on the right gives you a workflow how it is being used. As you can see, you can clone the protein of interest or the gene of interest into the vectors having large bit and high bit as a tag independently. Now, when these two plasmids are transfected into the cell, they make fusion proteins along with the protein of your interest. And if the protein A and B are interacting, then these subunits, they come together. As you can see from the image, you are not lysing the cells here, or you don't have a lysis step involved in this protocol. So we are going to talk about a little more how this assays make live cell detection capability useful and how it is going to enhance your research outcomes. On the other hand, NanoBret, where BRET stands for bioluminescence resonance energy transfer, uses a full-length Nanolux protein, which is used as a donor, and halotag is used as a receptor, as a acceptor molecule. The principle is almost similar to that of RET, which you must be quite aware of. So when protein A and B interact, the acceptor is to the donor signal. There is an increase in the acceptor is to donor signal. So more is the signal or more is the ratio, we can infer that more is the interaction happening between protein A and protein B. Well, there are a lot of techniques available at present which are used across the globe by scientists to study protein-protein interaction, like protein immunoprecipitation, ELISA, SPR. Then what is new about these techniques? What makes this technique more unique compared to what we have currently? Let's have a look at the highlights. The small size minimizes interference with normal protein function. The bright signal accommodates low native expression levels. The most important part, we can perform assays under live cell condition. So this assay gives you a platform where you can draw inference on protein-protein interaction at the physiological level. Many of you who are into research, they must have faced questions from reviewers that if the protein A and B are interacting that you are showing on an immunoprecipitation platform, how sure are you that there is a direct interaction happening? Or this interaction is because the interaction is happening during the time of incubation when you are preparing your antibody immobilized bits and adding them into the cell lysate and letting it incubate for around 45 minutes to 1 hour. So when exactly the interaction happening? Is it in a physiological condition or it is just a artifact? So questions to this answer can very well be answered when you use a platform where you are not lysing the cells and your inferences are from a live cell condition. Apart from that, you can perform real-time analysis of protein interaction dynamics as well. So this technology makes it very useful to be used for drug potency studies and binding kinetics. Besides, we can also use these techniques to monitor induction and inhibition of protein interaction. Apart from that, some few other features that make Nanolux a very ideal reporter is it is thermally stable, it is active over a broad pH range, and it doesn't require post-translational modification to work with. Well, luciferase enzymes are nothing new to viral research. These are used previously as well for making recombinant reporter viruses. These are nothing but are called as pseudotyped virus particles as well. So what is being done here is you are mocking the virus proteins which interact with the host receptor, and you are making the particles which is having a genetic reporter. The presence of this genetic reporter allows you to track the virus, and it also gives you a tool where you can do your viral research studies in BSL level 2. You don't need a well-designed virology lab in order to do such research. But in period of time, what has been observed by scientists is the compact genome doesn't tolerate large insertions. They get destabilized and they're lost over time. Or maybe because the replication or the packaging is affected. So scientists across the globe, they were in a hunt to find out a reporter which is small and which is sensitive enough that can be used for virus studies. To this purpose, HiBit has been successfully used, and this has actually helped the scientific community as there are publications or there are studies which shows that HiBit can be stably integrated into viral genome without disrupting the natural biology of virus. These virus particles are used in a variety of studies. For example, investigating small molecule inhibitors, investigating interaction inhibitors. Also, antibodies from plasma of recovered COVID-19 patients can be screened by using these viral research tools. And you'll be surprised or you'll be happy to know that the plasma therapy which is being given at present to the corona patients is one of such outcome from such studies. These are a list of publications which are new, and I would say they are they are more into the viral research and particularly into the SARS-CoV-2 research, where you can see various luciferase chemistry has been used from Promega into these studies. So you can refer to these studies to design your own assays. This research have given they will give you a very nice outlook or nice way out how you can design assays to question your hypothesis and prove that. Well, apart from the binding and apart from the normal course of action that the virus takes place inside the host cell also responds to the viral infection in a variety of ways. One of it is viral induced cytopathic effect. And the image on the left hand side bottom will show you one of the examples where viral tox flow can be used to study such effects. Apart from viral tox flow, Other products like cell titer grow from Promega can also be used. As you can see, compared to a control well where you have no viral infection, there is a test well where you have added certain amount of viral particles. And when you add viral particles, then they lead to cytopathic effect. And hence there is a depletion in the ATP level. This depletion in ATP can be correlated with a viral burden. This assay can also be further extrapolated to use in screening of antiviral drugs or antiviral molecules as well. So you can have your wells where you have your cells plated, and you can then you can add your viral titers to the cells along with the antiviral drugs. And if any of your drug is having antiviral activity or it has a very nice antiviral activity, then you will see the depletion of ATP will be least. Thus, it relates to the fact that when there is depletion of ATP is less, it means the infection is also less, and that particular drug is having the maximum antiviral activity. So, well, apart from this apart from the assays which can be used for cytopathic effect, there are assays from Promega where you can monitor nutrient uptake like glucose, and we can also we'll look for the changes in cofactors like NADP, NADPH glow is one of the assays for that. Glucose uptake glow is an assay where you can look at or where you can monitor the nutrient uptake levels in the host cells under viral infection. Well, we all know when a cell is under infection, the cell responds to it by expressing a variety of proteins, and hence the protein profile of a cell changes. Mass spectroscopy has proved itself as a very nice tool to study that. Trypsin lysine mix, one of the mass spec grade reagents from Promega, is used for in gel and in solution digestion of the cellular extract before you actually load that into the instrument to look at the protein profile. In the previous slide, we talked about a endpoint assay where we were using the cell titer glow or the viral tox glow to have a readout or have an antiviral drug screening. Now let's design a virtual assay and see how we can improve such assays using Promega chemistry. Let's say you have drug D1, D2, D3, and D4, and you are trying to study one time point to check out the antiviral activity of the drug. So this is how you plate the cells. You need 5 wells because you have 4 drugs and you have 1 control, and it is 1 time point. Now imagine a scenario where you want to do multiple time course. Let's have a look. Let's say you have 4 time points to study: 6 hour, 12, 24, and 36. So this is how the platform is going to look at if you are trying to do an assay. As you can see, you are increasing the number of wells. So you are handling more reagent and you are having more wells in order to get an inference out of it. So if I would say that Promega is providing you an opportunity or a tool that you can do all the time course studies, like all the 4 time course in the first format of your assay: 5 wells, 4 drugs, 4 time points. Yes, I am talking about a real-time course study of viability assays. In this assay, you are going to plate your cells in the media containing the real-time cell viability reagent. Then you add your drugs and let incubate for the time period that you want. And you can do a continuous luminescence reading of the same plate depending on the time point that you have thought about. In case you have an instrument which is capable of providing incubation parameters to your cells, then you can have the plate sit in the instrument. But in case you don't have that, then you can incubate your cells in a CO2 incubator, and depending on your time point, you can take that out and read it. So it makes your job easy by having only a single well where you can read your effect of the drug with respect to time. Now, how it is in another way, you can find this assay more useful, not just that it is reducing your uses of reagents. Let's say the drug is showing your effect somewhere in between 12 hours and 24 hours. But when you have designed your assay, you have set only 2 time points, 12 and 24. But your drug is showing the effect somewhere like maybe at 16 hours or 18 hours, but you don't have that time point in your studies. But when you try to do a continuous monitoring, you have a data at the 16th hour, you have a data at the 18th hour. So if some change is happening or your drug is showing your effect at that time point, you are able to capture that. So this is how a real-time assay makes your inference much easier and much valuable. Well, Promega not only just offers real-time assays for viability, it also has assays to study apoptosis and necrosis in real time. On the left, you have a basics of what is being used in this assay. As you can see, in case of healthy cells, the phosphatidylserine moieties are present in the inner leaflet of the plasma membrane. But when apoptosis is induced, there is a translocation of these phosphatidylserine molecules to the outer leaflet. And when you have your detection reagent is going to have annexin 5, which have been tagged with large bit and small bit, the same ones which you talked about in the protein-protein interaction. So this annexin 5 binds to the phosphatidylserine, which is present on the outer leaflet, and brings about the structural complementation of small bit and large bit, and hence you have a luminescent signal. On the other hand, the reagent is also having a DNA binding dye, which is a signal for necrosis. But as you know, during early apoptosis, you don't have disintegration of plasma membrane. So the dye sits outside the cell and it can't enter. So you only have a luminescence positive signal while the fluorescence is negative. In case of a secondary necrosis or in a time course where you will see that the apoptosis has now led to necrosis, then the DNA binding dyes, which is impermeable to the plasma membrane, can now enter the cell because the membrane has got disrupted, and you can see positive signal for luminescence as well as fluorescence. On the right, you have an image of that, a time course of course, as it is a real-time assay. You can see when the early apoptosis is induced, you don't see a fluorescent signal, but you see an increase in apoptotic signal from luminescence. Then, when the secondary necrosis is induced, there is an increase in fluorescent signal. That is because the binding of the DNA, it is because of the DNA binding dyes are now accessible to the nucleus, and you see the apoptosis finally is plateauing, and you see an increase in necrosis. So this real-time assays give you a very nice feedback to look at how exactly your drug is in, or what pathway your drug is following, whether it is following a apoptosis followed by a necrosis, or it is straight away following a necrotic pathway. Well, we talked about assaying viability in real time, assaying apoptosis, and assaying necrosis in real time. So let us talk about multiplexing such assays. So when we talk about multiplexing, it means you are able to read different parameters from the same well by sequentially adding the reagent one after the other. On the left, we have few graphs. Let's take a look at the graph of primary necrosis here. The viability is represented in blue, the cytotoxicity in green, and the apoptosis in black. You can see that the decrease in viability is caused due to the increase in cytotoxicity. Your apoptosis is not responding to the change. As you can see, the apoptosis graph is quite flat. Now, compared to that, if I look at the cytostasis graph here on the bottom on the left, then here the viability is represented in black, cytotoxicity in green, and apoptosis in purple. The decrease in viability here is because of the increase in apoptosis, and you don't see cytotoxicity responding because the graph is flat. So these assays can be performed independently, but when you do that independently, then you need to have 3 different plates, one for viability, one for cytotoxicity, and one for apoptosis. But when we multiplex, we can have a single plate and all the 3 parameters being read out. You need to add viability reagent, read it in a multimode reader, add the cytotoxicity reagent, then read it, add the apoptosis reagent, then read it. So you can see that it is making, or I would say it is making your job easy. In a single plate, you can do all these assays. Second, you are consuming less amount of reagents, and the third and the most important part is you are reducing the interval variability that would have caused due to manual error or due to pipetting errors. The figure on the right, which is a nice bubble chart, colorful, gives you a variety of assays which can be multiplexed. And if you have any questions on this, or if you want to design a multiplex assay which is going to fit to your requirement, we will be happy to help you with the reagents which are apt for your work. And do let us know if you have any such requirements. Now let's focus our attention to the immune response that is induced upon viral infection. The immune response can be adaptive or innate. In case of innate immune response, it involves the activation of inflammasomes and cytokine release. The activation of inflammasomes is quantitatively measured by detecting catalytically active caspase-1. But in case of COVID patients, you may have heard that the cytokine release is taking a form of a cytokine storm, and it has quite a bit of adverse effect in the patients. So doctors administer additional therapeutics to block the efficacy of the release cytokines such as IL-6. So from this part, we conclude that we need an assay where we can check the activation of inflammasomes for the release of cytokine, and we also need an assay where we can screen for therapeutics which can be used to block the adverse effect of cytokine storms. To check out activation of inflammasomes, Promega offers a tool known as the caspase glow inflammasome assay. Now, before we come to what assays can be done to check therapeutics or to screen therapeutics, I would like to highlight here that the therapeutics can be a small molecule or it can be a big molecule like an antibody. I guess you must have heard about antibody therapeutics, which are being used quite a bit these days. So in case your therapeutic is an antibody, Promega offers a variety of bioassays which can be used to screen them. So let's take an example of IL-6 bioassay. As you can see here, in case of bioassays, we offer you cell lines which has a luciferase cloned downstream to a response element. And this response element responds to a signal pathway which is generated when a ligand and receptor bind. In this case, IL-6 binds to the IL-6 receptor. It initiates a signaling cascade which is responded by the response element, and then it leads to more of luciferase production and more of bioluminescence. But if you have a drug, antibody therapeutic or an antibody drug, which is designed either against a IL-6 or it is designed against IL-6 receptor, either of them can block this interaction, and hence you will see a decrease or a depletion in the luminescence. So this gives you a very nice platform to screen high molecule or the antibody therapeutics. Well, bioassays are not the only tools that you have. We have developed another assay known as the Lumit immunoassay, which can also be used to screen small and large molecule inhibitors. Let's have a look how it works. In case of Lumit immunoassays, we tag the antibody with a large bit and small bit. Yes, these large bit and small bit are the same subunits of nanoluciferase which were used for studying protein-protein interaction. So in this case, we tag the primary antibody with large bit and small bit, and when the primary antibody binds to the analyte, that is the antibody which is designed against that analyte, when it binds to the analyte, it brings the large bit and small bit together. The structural complementation now brings about bioluminescence. This Lumit immunoassay can be done in 3 different formats. In direct format, you are labeling the primary antibodies. In indirect, you are labeling the secondary antibodies, and this makes this a universal approach to be used for any unlabeled primary antibodies. This assay can also be used to study competitive binding. And overall, these assays can be used for cytokine detection, metabolic target research, protein phosphorylation studies, and pathway analysis as well. Well, if you look at the entire workflow here, then it would more or less look similar to an ELISA. So now when we have an ELISA already in place, then what is the unique or the salient features of this immunoassay? Let's have a look how a ELISA looks like. So this this is a pictorial representation of a sandwich ELISA. As you can see, when we start with ELISA, we capture the primary antibodies, then we wash, block, then wash, then add the analyte, then wash, then followed by addition of primary antibodies, washing, secondary antibodies, washing, substrate addition, and this is when we generate the light, or this is when we generate the signal which we capture based on the reading platform that we have. In case of Lumit immunoassay, you need not do all these washing steps. It is not required. You just add your analyte, add your primary antibody. If your primary antibody are tagged with large bit and small bit, then you add your substrate and you generate the signal. In case your secondary antibodies are labeled with large bit and small bit, then you need to add the secondary antibodies, and upon addition of substrate, you generate a signal. So you can see it makes your workflow quite easy, and the time taken to generate the data is quite less. So you can perform an assay somewhere in between half an hour to 2 hours. And compared to that of the traditional ELISA that we have been currently doing, Lumit immunoassay gives you a very broad range to work with, and it also makes your job easy. So Promega has used this assay or this principle of Lumit immunoassay to develop antibody-based testing kits for COVID. And here I would like to play a video for you to better understand and visualize how it works. This will also give you an idea how the Lumit assay in general fits in for having a better detection. Let's look at the video.
The Lumit DX SARS-CoV-2 immunoassay is a novel system used to detect antibodies to the SARS-CoV-2 coronavirus. The assay is based on Lumit technology, in which proteins are labeled with complementary subunits of nanoluc luciferase, called nanobit, an engineered enzyme over 100 times brighter than traditional luciferase. Here's how it works. 2 fragments of the SARS-CoV-2 spike protein receptor binding domain are each labeled with nanobit proteins, large bit and small bit. The labeled proteins are incubated with diluted patient serum at room temperature for 30 minutes. If antibodies to SARS-CoV-2 are present, the large bit and small bit subunits are brought together to form a functional luciferase enzyme. The detection reagent is added and luminescence is produced within 3 minutes. Detect luminescence on a microplate reader such as the Glomax Navigator. Get results quickly at the push of a button using a preloaded protocol. The whole process, from sample to results, takes 1 hour or less. Detecting antibodies to SARS-CoV-2 is easy and reliable with the Lumit DX SARS-CoV-2 immunoassay.
Well, till now we talked about proteins quite a lot. Now let's change our attention to RNA molecules. Transcribed RNA are being used these days quite a lot for vaccine production. I guess you all must be aware of RNA vaccine these days. So in order to figure out the right RNA candidate, you need to screen a lot of RNA molecules, and this is where the transcribed RNA helps you in screening the right vaccine candidate. Apart from that, these transcribed RNAs can also be used to make recombinant virus particles. So these transcribed RNA are electroporated into the cell lines, and then you rescue the recombinant virus from the tissue culture medium after 1 or 3 days of electroporation. And these recombinant viral particles can now be used for a variety of research work. Apart from that, fish has also been used currently to study quite a bit of protein localization in specific tissues. And for that also you need transcribed RNAs. So Promega offers two main systems to make transcribed RNA. They are riboprobe system and ribomax system. Riboprobe system is used to produce radio-labeled RNA, unlike ribomax system is used to produce a lot of non-radio-labeled RNA, which can further be labeled according to your reading platform or according to your assay type. Well, now let's switch gears to surveillance in case of a viral epidemic. You must be aware of serological testings which are being done, and the outcome of the serological testings are being related to the how the population is responding, or a locality or the population in a locality is responding to the viral infection, whether there is an increase in infection or the infection has now stabilized or the infection is now going down. So a study by KWR Water Research Institute at Netherlands has shown that they could successfully find or successfully isolate coronavirus from wastewater. And they also figured out that the course of viral infection, the rise, plateauing of the infection, and the decline in that particular population could very nicely be correlated with the amount of RNA virus they could isolate from the wastewater. So this is where it gave an ideology that wastewater can also be used for epidemiological studies. So we at Promega scientists have developed two protocols to concentrate SARS-CoV-2 virus, one by using PEG precipitation and the other by centrifugal filtration. And then these viral concentrates can be used to extract RNA on automated or manual platforms. Promega is also developing amplification kits to detect SARS-CoV-2 in wastewater samples. What are the advantage if we use wastewater in these surveillance studies is it gives you an early warning. So before the onset of symptoms, you can see a viral RNA into wastewater, and that is what the study by the KWR Water Research Institute shows. It can be used for population-wide databasing, and it will give you a good idea what is happening in that particular locality, or what is happening in the locality where exactly all the wastewater is getting dumped. And the most important part is it is cost efficient. So well, this brings me to the end of the presentation that I had to share from my end, and I thank you all for your attention, and my sincere thanks to Biotecnika as well. So please stay safe and stay healthy, and please reach out to us and we'll be happy to help you. Thank you.
Okay, so thank you, Manas. So far, we have given an insights about different methodologies that can be used for coronavirus testing, and different technologies that can be used for COVID-19 vaccine research and therapeutic development areas. So in the changing eras of COVID-19 pandemic, the things are changing very rapidly, and technologies are keep changing, and the new things keeps coming. And it is really important for each one of you to get connected with us for receiving and more updates about what new technologies are coming from Promega sites and what are the new things and updates from us. So one of the ways to connect with us is the Promega's connection. What is in Promega's connection? Promega's connection is an online portal from where you can receive an information about different thoughts, technical tips, and different news from science from Promega Corporation. And in this Promega connections, we write a different block. You can access all the information online by just typing the Promega connection. But if you would like to get updated and see what new blocks each day we are writing and when it is coming, so you can also subscribe that, so that each time whenever new update is coming us, uh, so then you have an information at that particular point of time. So what you need to do is, on here in my screen you can see here there is a small icon where you can type your email address and click here for the subscribe. So you can search Promega connection online, and using these steps you can subscribe to Mega Connection for receiving an information about latest news and updates from Promega. In the Promega connections, there are also certain other things that might be helpful for you. So there is scientific resources available in the Promega connection. From that resources, you can have an information about different tools and handbook on protein biology, cell biology, and certain lab guides also. And there is also one of the very interesting thing you can find out in the Promega connection scientific resource page is the biomath calculator. So these biomath calculators are really important and helpful for those people who are doing a scientific experiment in a lab, and then generating a data and doing a lot of the scientific conversion. So these biomath calculators are designed to help automate some of your the conversions for the different experiments. So in the Promega connections, we have already written a lot of different blogs on COVID-19, and all these blogs are not only on the Promega's product, but some of the blogs are also on some of the things which is really important for COVID-19, starting from using, how to use the different safety measures in the lab when you start your work. So those kind of different generalized blogs are also available on the Promega connection. So once again, you need to subscribe that to receive the latest information. There is also a few other ways to get connected with us. So for example, like social media. So social media also, you can connect with us using the Twitters, LinkedIn and Facebook. So we have dedicated page on Twitter, LinkedIn and Facebook on the name of Promega India, where you can get connected with us and receive and newer and newer updates about what we are doing in a fight against the coronavirus, what new webinars are coming up. So you can receive all that information by subscribing to those pages. And you can also reach out to us directly. So you already have attended the presentation from us. And so here is the email IDs and the contact details available in this page. So if you have any kind of technical and even product related queries, you can reach out to us. On Promega's website also, if you can go to Promega's website, so you can also reach out to the respective salespersons for your respective territories and regions. Their details are also listed on the www.promega.com contact information. With that, I would like to thank you everyone for your precious time for attending this webinar. I would also like to thank you, Biotecnika, for giving us an opportunity and getting connected with all of you. With that, we are opening up for the Q&A. Let us know if you have any questions. Thank you, everyone.
All right, welcome back. So how was the session, everybody? Did you like it? Yes? Quickly in the chat box. Was that informative? I can see a lot of yes coming. Okay, fantastic. So welcome to the Q&A session, guys. We'll take a 10 seconds break and then we'll be back with the Q&A session. Now, how you're going to ask your questions? So you have to write a capital Q dot and then the question, okay? And that's how you're going to ask questions. We'll try to take as many questions as possible, okay? All our experts are right here, so to answer your queries. Okay, but however, it is possible that we may not be able to take up your question. Don't feel bad. There are so many of you. We may miss out, but yes, we'll try our best. So let's take a 10 seconds break and we'll be right back. Thank you. All right, so now we are ready for the Q&A. You can see live. We have all our experts right here. So we'll start with a couple of questions which have been asked previously in the session. Okay, so as you can see on your screens right now, the experts are right here. Start asking your questions. Okay, so I'll take up the first question, which was asked by Shubhankar Mohanty, that apart from ELISA or immunological assay test, is there any proteomic platform that can be used for COVID-19? So I would like to invite our experts to answer that query. Over to you.
Okay, thanks for the question. And that's a pretty interesting question here. So if we talk about detection, then we have either nucleic acid to target or we have proteins to target. So any type of assays where you target proteins, that can be used, not necessarily ELISA. And mostly for nucleic acids, being it is a RNA virus, you need to follow the RT-qPCR protocol. That's the best way we have today. So, well, this doesn't stop you from using any other protein assays. Yes, you can. You need to know the right target that you want to focus.
Okay, so I hope that answers your questions, Shubhankar. So the next question, which is in front of us, is how 400 samples can be tested on one day using Maxwell instrument?
Uh, well, I would like to clarify it. So, what Nitin wanted to present here, or what we wanted to share with you, that the instrument Maxwell RSC48 that can extract 48 samples at one time. So 400 samples uh, is like in a total shift of around 8 hours. Then you can isolate RNA from 400 samples, because in every go you have 48 samples. So you will run the instrument accordingly to have 400 samples being isolated. Well, now when it comes to RT-PCR, so we have two-step RT-PCR system as well as one-step also. So if we use a one-step PCR where the RT reaction and the qPCR, they are clubbed basically, or they are being done in the same tube, you just need to incubate the reagents according to the temperature requirement. Let's say in the first go you have to do a RT because it's a RNA virus. So you mix the reagents and then incubate it at the desired temperature for 30 minutes. Then when you are going to do a qPCR, shift the tube, or shift the tube to another platform where you can do the PCR. Now let's say you are having a 96 well plate format with you. So in one go you can do 96 well plate. So if you run 4 plates, then you are done with your 400 samples. So well, it completely depends what is the facility you are having in your laboratory, and what's the manpower, what is the workflow that we are following. And certainly during the COVID time, you are following a lot of safety measures also. So it is doable, but it all depends based on what facility and what instrumentation you are having at your lab.
All right. So I hope that answers your question. So now there is a question from Laishram. Why RT-PCR is giving best results as compared to any other serological test?
Okay, so I guess I guess you must have heard quite a bit about it in news as well. And so we had a lot of rapid tests in between, and they were stopped. So what basically, as a scientist, if you look at it, look at the scenario in a way that when there is infection, it's a RNA virus. So the first thing that you will be able to detect is a RNA. or to check that whether the presence of RNA or not, then you follow a RT-PCR technique. Once the RNA gets into the host cell system, then the viral proteins are being made. And we all know every platform has a window or a sweet spot in which you can detect that particular molecule. And when it comes to protein, you need to have quite a bit of proteins to be able to detect that. And also, the proteins basically that you are trying to detect are mostly the antibodies, and we all know that immunological response is initiated after some amount of after 1 week, some around 10 to 12 days post infection. So let's say you have a patient and you want to detect whether he's corona positive or not. The best way comes RT, because at that particular time, whether that patient is having infection or not, you'll be able to check that. Now, if you go to the antibody testing, then either the infection should have been about 5 to 6 days or more than that. So it is important that how you correlate the data that you get. And if you don't, as a scientist, you need to have a pattern, and every experiment, every studies, they have a biological pattern to follow. We cannot change the biological pattern. So that is why RT-PCR becomes a good technique to instantly get to know whether there is an infection there or not.
All right. So I hope that answers your question, Laishram. So there's another question coming in. Antibody testing does not give accurate results at the initial stage of infection. So why it is not very effective? That's the question.
Well, I guess we talked about it just now. So when you have an infection, your host cell responds to that and produces antibody. So the body takes about 1 week, 10 days or 12 days. Now, depending on what is the number of days of infection, you have IgG or IgGm being developed. So if you are not testing your if you are using an antibody testing kit post that much time of infection, then you will get a good better result. But let's say you are doing the testing in the first 3 or 4 days of the infection, certainly the body has not developed antibody, so you will not catch it up in a test kit.
All right. So I hope that answers your question. Now, as I can see, there are 120 plus questions coming in. Guys, we will not be able to take all the questions. We'll take some of them. Please don't mind. So the next question, which is there from Kabita, and she says, how rapidly the spike of nCoV-19 virus changes? So how rapidly the spike protein changes? That's a question.
Uh, well, that's a pretty interesting question, I would say, and very nice question for this hour when we are actually going through a pandemic. But yes, we are not viral experts, so this question can be better addressed by a viral expert. And the tool that you can use to do that is to check whether there is genetic mutation happening, and if those mutations are bringing about a functional change into the proteins, and whether the functional proteins are spike proteins or not. There can be any proteins, because virus always tries to take a route so that they can escape the host immune system or escape the host surveillance system or the radar system, that you can understand it better. So that would I think that question can be best answered by a virologist, and we are not experts in that.
Right.
Well,
Okay. So I have a quick, very interesting question coming in. I guess Nitin or Rajnish can also come in on that question from Devjani. And she asks how Promega uses its techniques in determining different strains of SARS-CoV-2 virus or differentiate between the mutated ones.
So, well, I think the techniques which I think we have covered at the beginning is basically the recommended techniques from the ICMR. We basically comes up in the pictures for supplying the reagents and components. And again, I think I would say the scientists and the virologists are the experts to determine in that areas to suggest that what techniques would suited in that particular scenario.
All right. So I hope that answers your question. We have a next question coming in from Kavya Shri. Why we use Tachman probe in RT-PCR and what is its importance?
Okay. So when we do a probe-based detection, probes can be of different types. Tachman probe is one of the many types which is being used, and that is the most widely used probe, I would say. And when we use a probe-based detection system, then it makes your assay more sensitive and more specific. Now, apart from probe-based, what you have as a detection platform is a dye-based. So when we use a dye can bind to DNA. So any DNA that is getting amplified, dye is going to bind to that. It is not having a specificity for a particular DNA strand that you are looking at. So let's say during your amplification, you had a non-specific amplification going in the background. The dye is also going to bind to that, and it is going to give you a signal to that. But that is what you don't want. So when you do a Tachman probe, you use a quencher and a fluorophore in combination. So if you try to look at the workflow of a Tachman probe, then you will see that it adds more specificity to the template that you are targeting. That is why Tachman probes is being used in RT-PCR, and that makes it more specific. And we all know that when it is PCR, you amplify so many number of copies. And if you have a small one template also, that will amplify and that will going to give you some signal, and that is what you don't want. So we want to have a signal which is specifically coming out from the template that you are targeting. So that's the answer to the question.
All right. So the next question is from Shabana Kadri. What is your opinion on the upcoming vaccines?
uh, well, so we are all looking forward to have a cure, and vaccines. We all know that is the best cure. Why? Because it is not targeted only to the patients who have viral infection or COVID infection. They're also targeted to the common man, because we all know today that we have a lot of asymptomatic cases going on. But now, when it comes to vaccine, you need to have the right candidate being injected. That is very important. It is not just to have a vaccine. It is very important to have the right vaccine, because you can't just inject any molecule to have a immunogenic response that may not work for you also. And when we do a screening of vaccine, there are multiple stages that one goes through. If we look at the recent news, few of the vaccines who have been successful in the trials, they are being tested with the patients who have infection. But the target of a vaccine is not only those. The target of a vaccine is the common man as well. They may or may not have infection. So now when you do a bit more of human trials, then we'll get to figure out, okay, this is the right candidate, and that's what we should wait for. And till that time, I think we should take all the hygienes, and we should take all the safety precautions rather than being panicking that okay, what is going to happen, or rather than thinking that okay, when we have the vaccine things will be okay. No, it's not like that. If you because if you have already developed immunity that we are calling a herd immunity these days, so you are fine. That is completely okay. You don't have to pay for the vaccine now. You are already immune. So let's take it on a positive note, and we stay safe by following all the hygienes, and hope that we have a vaccine soon.
All right. So our next questions are flowing in. I can see that. So there's a question from Saili. I hope I'm pronouncing your name right. So Saili says that all the techniques used for detection of COVID-19 gives false positive results also. If yes, then how much is the false positive?
Okay, so let me take you back again to the course that we discussed. Most of the tests, either they are going to use antibody as a target molecule, or a protein as a target molecule, or a nucleic acid as a target molecule. Now, we design a test kit by using primers, which is going to bind to a specific, which is going to bind specifically to the corona virus. Now, not necessarily whatever primers we use, that is only and only going to bind to that template. As I guess most of you may be into your BSc, MSc, or into your PhD, we all know when we try to design an in vitro assay, then a lot depends what is the amount of template present and what is the amount of primer, what is the formulation we have inside the tube. So all these things matter. So yes, you can have false positive results as well. And which kit has what is the which kit has what extent of false positive that is being tested before it is being used in the diagnosis, and that comes under the quality control of any kit. So so whatever kits are being used today, they are being first quality assured and quality screened before they are being used uh, in the public for testing. So so I would say it doesn't have a definite answer. You may have variability across kit to kit, but whatever is being used, they have quite a bit of minimum false positivity, I would say.
All right. A lot of questions flowing in. Okay. There's a question from, I guess, Sunil says that is Promega having any expansion plans further in India? So I guess,
I'll take this
Well, I guess
question.
Rajneesh.
yes.
Yeah, I'll take this question. Uh, to be very honest, we are still evaluating the situation, and as on date, we do not have any plans of doing any manufacturing. We'll continue with our sales and marketing activity, what we are, because so far the logistics are so strong in Promega that they are based in US, and we get the shipment, we get the R&D product, we get the solutions in hardly about 2 to 3 days turnaround time. So my answer to you, as on date, we don't have any plans to go into any other activity other than sales and marketing and application support, which we are doing to all our customers.
All right. So there's the next question coming in quickly. I believe again relates to you. So if somebody has to buy Promega's products, whom should they approach, and how should they approach?
Okay, well, I think let me answer as from first-hand
Yeah.
information. So I think the first point of contact is like if somebody is totally unaware about how to contact, so anybody can reach out to www.promega.com, and we have listed the email address, and in fact, these days we have also put it up the contact informations also from our office contact numbers or even the contact numbers of our sales team, so they can probably start it from there. So there is a one email id is called ind cursor, so probably they can write their inquiries there, and we can direct that queries to the respective channel partners or salesperson.
All right. So there you have it. If you want to buy any product of Promega, you can visit their website and check out the contact details, and they will be right there to support you. Okay. One last question we have. So this is the last question we'll take, guys. Okay. So one question from Linda Johnson. To which part of the antibody does the Lumit bind to make luminescence? So I believe Manas can answer that.
So may I have the question again, please, Sekha?
Yeah. So the question is, which part of the antibody does this Lumit bind to give luminescence?
Okay. So when we design limit assays, the large bit and small bit, they are tagged at the constant region of the antibodies. So the FAB part of the antibody is what it targets to the analyte, and in the limit immunoassays for SARS-CoV-2, it is designed against the viral proteins, and the FC region is what is being tagged with the large bit and small bit.
All right. So I hope that answers your question, Linda. So now there has been a repetitive request that they wanted the PowerPoint of this webinar. Will that be possible? If you can let
Yes,
us know.
yes, definitely. We will take some time, and we will, I think, coordinate with you guys to make sure that
Yeah.
the
All right. If
important
that's possible, because
information reach out to everyone. And
yes,
plus, in the
yes,
meantime, I think we have already shared the some web link, because all the information whatever we
yes.
have presented is there on our website as well. So everyone
Yeah.
can go to www.promega.com, and there they can find out two page. One is for the COVID-19 vaccine research, and one is for the therapeutic areas and the COVID-19 testing. So they can reach out to there also to find out the entire information. And in the meantime, we will work to ensure that important information reach out to everyone.
All right. All right, guys. So there you have it. You heard from the experts. If you want to reach out, they've shared their email IDs. You can reach out to Promega, okay, on the website, okay. And with this, we come to an end of our session. Thank you so much, everybody, for joining in. Thank you, Promega team, for being there for us, and you joined us this morning. Of course, you have a very busy schedule. Still, you took out time. I'm really, really thankful to Manas, Nitin, and of course, my dear friend, Dr. Rajneesh. Thank you so much for joining in. So, with this, guys, we come to an end of our session, but don't go away without filling the feedback form. Now, your feedback is very important for us. I personally read each feedback and see how can we improve our next webinar. And yes, if you are filling the feedback form, since you have attended this webinar till the end, you are going to get the e-certificate, participation certificate jointly from Promega as well as Biotecnika and Rasayanika. My heart goes out to all of you. Thank you so much for joining in. I know you all are very busy people. All the biotechnicans out there, keep shining. Okay. The feedback form will be shared with you now. Okay. Let me quickly do that to all of you. You should be getting that right here. Now, once you have filled the feedback form, you will be redirected to a surprise gift from my side, which you can obviously encash. Okay. And yes, the certificates, the e-certificates will be given to all of you in the next 7 working days. Thank you so much, everybody. Keep shining. We will meet you again soon in our next webinar. No, that's not the announcement. We are going to conduct an online poster presentation in our next announcement, which will be there on the website very soon. And that will be jointly by our co-organizers, ABLE and KITS, Karnataka government. And that will be announced shortly today. So stay tuned. Do not forget to register for that, because there are prizes from IIC Bangalore, Stemputics, IBAB. All of them are joining us for this wonderful poster presentation online. Thank you so much, everyone. See you soon in our next one. Thank you. Bye-bye. Have a nice day.